Yeah, thank you so much for your, for accepting the invitation to give us a talk. So first, let's give a short introduction about Professor Huang. So Professor Huang is the deputy head of the Department of Food Science and Technology from National University of Singapore, and also the director of Biomedical and Health Technology Innovation Platform of NUS Suzhou Research Institute. His main research interests include chemistry of food bioactive components, design of sustainable staple foods for health promotion, alternative proteins from plant microalgae, and cellular agriculture. So Professor Huang had obtained a PhD from Indiana University and did postdoc training at MIT. So today his topic will be impact plant proteins on the texture and in vitro digestibility of surimi gels towards high protein foods for consumers with dysphasia. So now the time is yours, Professor Huang.
Thank you so much, Professor Suen, for
Uh,
giving me this opportunity to present. Actually, I'm not a protein nutritionist, so I have learned a great deal from previous speakers talking about very beautiful work on these peptides and proteins in animal model studies. And in my lab, we do much animal study on protein nutrition, but we do research on phytochemicals in animal models. So today my talk will be a little different. We are going to talk about how to formulate protein food for elderly people who have swallowing difficulty. As I told Chen Chai before I accept this presentation, actually I didn't have much of any more model to talk about. So I want to do some advertisement, which is actually recruitment of our faculty member in National University of Singapore. This page is actually the university town of our NUS, which is a very beautiful place. And I think so our department, we are recruiting many faculty members. Uh, Actually, we have been doing so for a number of years. Many of the, I saw some familiar names on today's audience, and I think, as you know, NUS have been recruiting, and but it's very difficult to attract people to Singapore somehow. And I think maybe people think it's quite difficult to get anywhere. I think in the past there were a period which our tenure promotion is quite tough. This year's I think the successful rate is much higher. So I think uh, for those who really have uh, keen interest to relocate to Singapore, as a full professor, associate professor, or assistant professors, we have like many openings still. We only expand our department to 20 faculty members. And if you have a postdoc or senior PhD students, you might pass this information to them to actually check out on our website. We have all the details. And okay, actually, we are also hiring a director for our resources center, which started September last year. And this one is really on plotting because the resources center for sustainable plotting is actually multidisciplinary teamwork composed of over 20 principal investigators in Singapore as well as internationally. And the mission of this Bezos Center for Sustainable Floating is to spearhead leading-edge interdisciplinary research and innovation to accelerate the commercialization of ultimate proteins. It's quite ambitious. Technologically, this proteins will have superior shape, hybrid, which is basically the hybrid proteins in particular. and that is the price parity to the traditional meat-based ones, and tailor the taste and nutrition for diversity of consumers. I think it's a very long sentence, but that is what we try to tackle. And today's work falls into this category as well. We try to develop hybrid food by using plant protein in combination with the fish protein to achieve the desired texture profiles as well as the digestibilities, but we only look at it at in vitro digestions. And I will jump over my research group. We don't do much protein. The previous work mainly on small molecule, phytochemicals, and until in the past few years, we look into proteins on the food processing, extraction, formulation to the food that people like to consume. We didn't do enough on the nutrition aspect of the products we developed yet. And but we actually are in the business of this protein because as the population grows, the demand for protein increases tremendously, and as well as the aging societies, as you know, Singapore is already an aging country, and so we need food not only nutritious but suitable for elderly citizens to consume. That is the challenge we want to tackle. So for all the folks, the problem is the swallowing difficulty because the muscle wastage happen when we grow old. And so depending on the theories of the swallowing difficulty, we can actually group them into different stages as wait, let me put a pointer. Basically the key parameter to define if the food is suitable for the elderly to swallow or not can be quantified by the hardness, and so as the swallowing difficulty increases, the elderly people can only swallow very soft food with hardness is like 10 to the power uh, 3, 5,000. But there were other disease, like patients who may also suffer from this swallowing difficulty, including those who may have health conditions like cancer or other issues. Yeah, so actually, to develop a special food with high protein content to meet this special dietary need consumers are very important. And so we look at surimi because surimi is one of the largest protein source, and it is certainly a complete amino acid and excellent protein. But surimi, and also surimi is actually the fish protein that's removed from the fish so that there is no bones or no other uh, hard materials. So it's a very good protein-rich food that can be formulated into different kind of fish uh, products, including fish ball and so on and so forth. But the fish proteins is mainly myofibril protein, which is very different from the plant proteins that is globular most of the time. And so in this surimi, and we can actually form these surimi gels, which is in the form like fish ball or fish cake, and very high protein content by just simply blending the surimi with salt. And so it can actually self-assembled in a sense through 2 stages or a low temperature and then heating it up so that it is set into the final form. And so the interactions among the surimi protein chains could be mainly hydrophobic, and also this gamma glutamyl linkages. I don't know how much gamma glutamyl peptide in the fish meat. Maybe it is quite significant because the fish certainly taste a lot umami than the plant proteins. And but to make it soft, then what can we do? And we actually uh, can do a number of things. For example, you can use different ingredients, which could be the additives like different salts, like calcium, or could be adding some fat, lipid, for example, and which can actually alter the surimi gelation textures a little bit, as well as this carbohydrate, soluble fibers, so oligosaccharides, fucoidan, and so on and so forth. So we can actually trick this texture properties and organoleptic attributes for the final product. But the main issue is to make it softer. And so how can we do that? We actually can look into a number of tricks which we can do by actually let me jump into this slide, so you don't have to wait for too long for your lunch or go to bed. In this part of the world is almost midnight, and I'm hitting in this Penang in Malaysia. I just arrived 2 hours ago. And yeah, so how can I make it a surimi gels soft so that it is actually suitable for elderly people to consume? We can use enzyme, for example, like papain, bromelain, or we can actually mix in like plant proteins, like soya, pea protein isolate. And certainly, more recently, the very hot research topic is to add this microalgae extract, which is plant protein, microalgae protein enriched powder. And so all this have been demonstrated to result in the soft surimi gel. And this work, I forgot to mention, is by one of my PhD students, Jane Lee. And she has done all these different methods and all end up with a softened surimi gel, which is suitable from the not in human clinical study, but from the texture profile analysis. And I think it looks promising. So the plant proteins, we want to mention a little bit. This soya and pea protein isolates are the most common uh, products sold on the market, and it basically has high globular structure, and the gels formed from these proteins are typically very soft, or could be too soft and mushy and won't form a good shape unless you add coagulating agents, for example, like calcium, magnesium, and so on and so forth. And and also plant proteins has its deficiencies in certain amino acid. For example, like a sulfur-containing amino acid like methionine or cysteine. And but the negative impact also include the typically the plant proteins have some color associated to it. Could be from the residual pigment get absorbed in the protein, unlike the surimi, which is purely white color. And so when you blend plant protein with fish protein, you may actually get some products which not as white as the pure surimi based product. The other negative aspect is actually this beanie flavor. And this beanie flavor is very strong in soya and pea protein. And but in this part of the world, like in China or East Asia, people got used to the beanie flavor products, including like soya milk, And actually, people prefer the soya milk with beanie flavor. In the US, on the other hand, the consumers do like this beanie flavor. And you can buy this uh, 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 soya milk from, for example, Trader Joe. They sell the soya milk without any beanie flavor. I don't know how they remove the beanie flavor very cleanly. Maybe they use a special type of soya bean to do that. And so when we actually mix this soya protein with uh, the surimi, the good thing is the surimi came with a very nice fishy flavor, and that can perfectly mask this beanie flavor. And so the hybrid protein gel has a better flavor attribute. So how do we do the making? It's quite simple. It's just mixing this surimi we bought from the local fish ball company and blend it with the soya or pea protein and add some salt. And then we let it set for 30 minutes at 40 degree and for it to form gel. After the gel is formed, then we cook it 90 degree 15 minutes. And that is basically the final product the consumer can consume, which is mixed surimi and protein and plain protein gels. And so we then characterize particularly the texture aspect of it, as well as the in vitro digestibility. And before we look at all the textures, you see, as I mentioned, this SPI, soya protein isolate, and P protein isolate, as you blend in different percentage, like 20, 40, 60, 80 percentage of the plant proteins, the color become darker and darker, and which is may be an issue for certain consumers who may prefer certain colors. But that is very difficult to avoid at this moment, unless we can isolate purely white plant proteins. Maybe in the future we can trust rice protein, which are almost colorless. And that's the color issue. Then on the gel properties, we did the rheological measurement. And again, it actually, the gel seems to be still a lot elastic and instead of viscous systems, as a G prime is larger than G double prime. And so it does form this gel network. But we actually observed that this G prime and G double prime is actually correlated to the percentage of the plant proteins we're adding. And in the beginning, as you see, this is G prime actually decrease, and then then when you actually at 60%, somehow it peaks tremendously. And for the soya protein and for the pea proteins, it peaks at 80% of the pea protein. So, but one thing you can actually see is you can tweak the texture profiles by using different ratios of the plant protein and the surimi proteins. And we also measured this creep and recovery, which also shows this that is some peak in 60 and 80% for pea plotting again. And so this may indicate some sort of a molecular interactions, but certainly this rheological measurement won't tell us too much. And so we did this gel profile studies during the formation, like incubation in the first 40 minutes, 30 minutes at 40 degree. And then we ramp the temperature up to 90 degree cooking. And so when the gel is actually developing, the rheological change is quite minimal until actually after we heat it up, and then the gel actually hardens. And so our understanding is actually during this denaturalization cooking process, there is a more cross-linking formed among the proteins, but we still do know how these globular plain proteins interact with this surimi proteins. And then we run SDS page. And from this SDS page, you can actually see there is not much of a change in terms of new band. Basically, which it means there is no chemical bonding interactions between the plant proteins and the surimi proteins, and which is understandable. I think this plant proteins lack like a thiol, like a thiol containing amino acids, which may form disulfide bond with the surimi protein. And they basically are mixing in a physical interactions at the most hydrogen bonding, hydrophobic interactions, and so on and so forth. And from this protein structure analysis, actually this bonding analysis, we see very little change as well. So which is indicating it will really cause a lot of conformational changes of this surimi as well as the plant proteins. So then what is actually the interactions happening uh, to actually change the textures? What we are proposing is basically they just blend into mixtures, and so that the originally very densely packed surimi gels is loosened up, and which can be easily seen from this confocal microscope as well as this SEM. And in the beginning, so when you don't have any plant proteins, you see this nicely packed surimi protein both in this confocal as well as in the SEM image. But when you increase the amount of plant proteins you added in, you see this plant protein yellow dots basically start to spread around this surimi proteins, and eventually the network is disintegrated and into like a particle form. And I think that is basically an easy explanation of these interactions between plant proteins and surimi proteins. We did texture analysis of the cells, and then we did actually also found inconsistent with our visual observations. You see this surimi, the gels contain this soya protein isolate or pea protein isolate, and it become much more mushy and moist and adhesive, granny, instead of springy and hard and cohesive, which is the sample that have no soya proteins in it. And so that is basically the end result. And then we did some in vitro digestibility test of this mixed gel, and which shows the digestibility is still quite good, although it did decrease a little bit percent of free amino acids, but not much. Yeah. So in a sense, the mixture gels does not negatively impact the digestibility of the proteins, and in at least in the in vitro experiment, and which shows in both the soya protein as well as the pea protein samples. And so what is actually happening? So it's quite simple. I think our story is just like when you mix this surimi protein, the fibrous with the plant protein, globular proteins, and their interaction is basically the globular plant proteins disrupt the otherwise densely packed surimi fish proteins, and it form this kind of aggregate which actually have a loose intermolecular interactions and softens the gel. And also it may disrupt this disulfide bond interactions a little bit, but the evidence is not very conclusive. Yeah, so to cut the story short, I think we have successfully developed this hybrid gel by using soya protein and pea protein isolate with surimi, and we can actually soften the hardness of the gel so that it become moist, adhesive, glanny, and mushy, and suitable for the consumers who have swallowing difficulty. But due to the constraints, it is extremely challenging to conduct the human clinical studies. That's the dilemma because to gather the data on human, we would have to recruit the patients who have the swallowing difficulty, and which it itself poses a great risk because if something happens, and then we are in big trouble, right? So I think I don't know how this can move on, but somebody volunteers have to try it. That's the difficult part when we do research on the food on the elderly people who have swallowing difficulty. And so we also tried these microalgae proteins, but I think due to the time constraint, I would just stop this part of the story. Yeah, I really thank my big research group, but the majority of them are working on like a cellular agriculture, plant plotting, 3D printing, bioactive molecules, and a lot of them working on food chemistry. So we don't do much on nutrition, unfortunately. We wish we could do more. Maybe we can collaborate with some of you to conduct animal studies on this plant protein nutrition. I want to thank a lot of collaborators over the past years, but most importantly, I want to thank funding agency, particularly Singapore Food Agency, Amway, China, and of course Bezos Fund, Bezos Earth Fund for actually setting up a center in our department. Thank you very much.
Yeah. Yeah, thank you very much for Professor Huang's nice presentation about using like plant proteins to improve the like the soft gels of the surimi gels, make it like better for the maybe for the elder people, right? So a very nice presentation and very nice work. So do you have any questions for Professor Huang?
Thank you, Chen Cai, for your good
Yeah,
words.
thank you. Yeah. Um, um, so I think one question is, do you think that if we combine the plant proteins with this like fish remi gels, will that potentially like improve their digestibility and also improve the like the gut health? Is there any like previous reports or any clues about it?
Yeah, that's a very good question. Actually, this plant proteins, it may not be pure, actually, may also contain soluble fiber in it. And
Mm,
so that may help in terms of the prebiotic property for the gut health.
mm,
And particularly for this microalgae protein powder, and the
mm.
protein content in that is less than 60%. And then the soluble fiber content is actually quite significant. And so, as a previous talk, we also discussed a bit of this interactions of the this carbohydrate, not carbohydrate, but this glycolated proteins in the gut for the gut health. It is
Mhm.
certainly can improve the I think the mixed gel using these plant sources, which may contain soluble fiber, or we can purposefully blend in some soluble fiber
Mhm.
as well. And that will improve the gut health aspect. In terms of the digestibility, we in vitro digestion didn't seem to have much difference and according to what we observe. But we certainly need to do more work in
Mhm.
animal model to evaluate the digestibility and amino acid scores.
Okay, yeah, thank you
And
very much.
from the interactions, I think molecular level, there is not much of a cross linking or anything, which may have an impact on digestibility.
Okay, I got it. Thank you. I think Donald, please, yeah, please unmute and ask questions.
Oh, hi. Dr. Wang, very interesting studies.
Thank
So
you.
I want to ask you uh, two questions.
Yeah.
Why do you choose surimi as a product to be modified by the pea proteins? And the second is surimi traditionally is a Japanese oriented food. How popular or is there any significant industry in Singapore or Southeast Asia about the fish surimi? Thank you.
Yeah, that's a great question, Donald. Yeah, actually, surimi is a extremely popular in Singapore. And actually, Singapore don't make a lot of food, but fish ball is one of the item that Singapore has even have a national standard. And Singapore, the fish ball company, actually sell the fish ball all over the world. So it's a big industry in Singapore. And because Singapore, we got the surimi from the neighboring country like uh, Indonesia, Vietnam, and Malaysia, Thailand. We do actually have a fishbowl company who are doing very good business on this. And fishball is actually very common in Singapore. And I think not just in Singapore, in Southeast Asia, is a very popular item. If you come to Singapore, you see lots of fishball noodle, fishball in like in hot pot, fishball. I think due to its, I think uh, the taste is great, and also it has no fish bones, and that could be the reason why it's so popular. Yeah. I don't know if I answer your question, but it's quite hard because it is bouncy like ping pong ball. When you eat hot pot, the fish ball drop on the floor, it can bounce back.
So what
That's
are the
very fun.
major fish you over there to use to make a fishball or a surimi type of product?
There were a number of fishes. I couldn't remember what particularly fish they use. Typically, they use a mixture of different fishes, at least maybe two different fishes. I couldn't remember on my head what fish they use at this moment.
Oh, thank you. Yep.
You're welcome.
Yeah, thank you.
Dr. Huang, if you don't mind, I ask a very naive question.
Oh,
Um,
yeah, yeah, please go ahead. Yeah,
yeah, so
no naive question. Yeah.
no, yeah, so like say, I was wondering if there's any other. I mean, because in your study, you say you suddenly you plan to protein, like say pea proteins and with surimi. So what about like,
Yeah.
is there any are there any other techniques to process a surimi or other type of foods for people with swallowing difficulties or dysphagia?
Of course, there are other type of food, and we are actually developing this soft. We want to develop lot this this high protein food, which I think, and also delicious food. And there were, of course, the traditionally to soften any high protein food. Very often we use enzyme like protease partially digest the meat, or could be using some fermentation. And but of course, fermentation will actually cause some flavor issue. And yeah, so the direct blending of the plain protein with the surimi is a very simple method to make a very soft, high protein fish with a good taste. Yeah,
Yep. Thank you.
welcome.
Yeah, thank you very much. y- yeah, if no more questions, maybe Hongbin, should you wrap up the?
Yeah, yeah. I think,
Okay.
I guess, some people have already left, because like, somehow, this is also very late, especially in China, in Singapore, in Asian areas.
Yeah.
So, we thanks everybody for joining us today. We try to accommodate time for different time zones. We know like it does not work perfectly. So, I wish everyone, if you're in China or in Singapore, so a good night, and have a good rest of the weekend. And also, if you're in North America, have a good rest of the day and have a good weekend. And we will post these videos on our CAF's YouTube channels. So feel free to check if you like to see more the presentations, or you like to communicate with the presenters. And we hope everybody enjoyed the presentation, and thank you for joining us.
Yeah, thank you everyone for joining us and
Thank you.
give a
Thank
good
you, Prof.
presentation and
Huang.
thing. Yeah,
Thank
thank you
you.
for the time.
Thank you, Chen
Yeah,
Chai.
thank you. Yeah, thank you,
Okay,
Professor Huang.
bye-bye.
Bye.
Bye-bye.